Hello, and welcome to the November Research Information Volunteer Call on disease-modifying therapies for type 1 diabetes. Today's presenters are Marinac, Senior Director of Regulatory Affairs, and Frank Martin, Senior Director of Research.
Hi, I'm Frank, and it's my pleasure today to speak to you about two of our major research priorities in our cures area. I'll be talking to you about our global universal screening initiatives and our disease-modifying therapies initiatives. So, what are disease-modifying therapies? Disease-modifying therapies are therapies that are going to preserve and restore your own pancreas's ability to produce insulin. Because in a healthy pancreas, your beta cells are the cells that produce insulin, the hormone you need that will allow glucose to enter the cells in your body and power them, give them the energy they need to function. However, in type 1 diabetes, your immune system has decided to turn onto itself, turn onto your body and destroy those insulin-producing beta cells. This causes glucose levels to increase in your blood, and then you're going to need insulin treatment to survive. You're going to need to inject or deliver insulin to survive. So in type 1 diabetes, we want to be able to provide disease-modifying therapies to either prevent, halt, or reverse the loss of those insulin-producing beta cells. This is going to change the course of the disease, and it's aimed at every person with type 1 diabetes at every stage of the disease. These therapies are going to be directed towards either the immune cells in those people or the beta cells in those people, or both. Now, let me remind you, type 1 diabetes has a number of different stages, and type 1 diabetes does not start the day you're diagnosed with type 1. It starts many years before. It starts when your body starts producing signs that it has autoimmunity. Those signs we can read by testing your blood for something called autoantibodies. These autoantibodies are present when you don't have symptoms of type 1 diabetes. You don't feel highs, you don't feel lows, you don't feel any different. We call this stage 1, when you have normal blood sugar and autoantibodies circulating in your blood, specifically 2 or more of the type 1 diabetes specific autoantibodies. Now, when people progress from stage 1 to stage 2, their insulin producing beta cells have started to function poorly, or they've started to die. But these people are still not feeling symptoms from type 1 diabetes. The only way we know they've progressed from stage 1 to stage 2 is when they go into their doctor's office and their doctor does specific metabolic testing. This is stage 2 disease. Then people will progress from stage 2 to stage 3 disease. This is when a significant number of their insulin-producing beta cells have been destroyed by their immune system. This is when people need insulin to survive. Their body can no longer control the amount of blood sugar in their bodies. This is classically when people are diagnosed with type 1 diabetes. And then people move into stage 4 disease. This is what we call established disease. This is when people have begun to sort of stabilize in their disease after that very volatile first year or so after diagnosis. So for our cures area, specifically in the disease-modifying therapies approaches, we have 2 main strategies. One is we need to turn off the autoimmune attack against your own insulin-producing beta cells. We need to stop your autoimmunity because that's going to keep killing your insulin-producing beta cells. Second, we need to be able to sustain the survival of those beta cells, and we need to be able to create new ones in your own body. We need to be able to coax your remaining beta cells to multiply, to replicate. So our disease-modifying therapies area will include treatments that's going to both control your autoimmunity and preserve or regrow your beta cells. And JDRF is accelerating the development of these therapies from the very earliest stages of discovery research all the way into clinical testing, and eventually towards regulatory approval and access. So part and parcel to our CURES program and integral to those efforts is our work around T1D risk screening. Why do we screen for type 1 diabetes? A very dangerous event can occur when people are being diagnosed with type 1 diabetes called diabetic ketoacidosis. This is when the blood sugar gets so high that it starts causing damage to other organs in their body. And this can lead to people ending up into the hospital or having very serious organ damage. Unfortunately, this occurs in about 60% of people who are being diagnosed with type 1 diabetes, especially those people in the general population who don't know that they have a family history of the disease. So 60% of people are being diagnosed with type 1 diabetes during this serious hospital occurrence of a DKA. However, when we provide these people with the ability to get screened for their risk of type 1 diabetes and also provide them with the requisite monitoring programs after they know their risk status, we can reduce the risk of DKA at diagnosis to as low as 3%. Why is this important? We know that if we reduce the risk of DKA at diagnosis and reduce those rates, those people are going to have better near and long-term glucose control, lower HbA1c, and lower complications of type 1 diabetes, also improve cognitive function. So that's just one reason why screening is important. Another reason screening is important is because the clinical trials we run to test the drugs that we think are going to have potential to cure type 1 diabetes are dependent on the participation of people who are either at risk for the disease or recently diagnosed for the disease. We need to be able to find these people and encourage them to participate in these clinical trials to accelerate the development of these therapies. So if we can increase the number of people screened, we can increase the number of people who are potentially able to participate in clinical trials, or more importantly, can benefit from those therapies when they're approved. Now, the current T1D screening programs are actually based in the familial setting. They screen people who are first-degree family members. So our aim is to capture more of the people who are at risk for T1D. Familial screening programs only capture about 7% of the people who are actually at risk for the disease. That means we're missing 93% of people who are at risk for T1D. Also, most screening programs are focused on the pediatric community. 50% of T1D diagnoses are occurring in the adult setting. So we're also missing those adults who are being diagnosed with T1D. So JDRF's approach to screening is to support research studies in the pediatric and adult setting that give us the body of evidence we need to facilitate policy development so that we can have T1D risk screening embedded in our healthcare systems here in the United States and across the world. And we believe we need to support general population screening efforts to better capture everyone who's at risk for the disease, not just those people who have a family history of the disease. So in the last 10, 20 years, so much success, so much benefit to T1D families has been found through programs like T1D TrialNet, through India, through Baby Diab, these familial screening programs that have studied the changes in the at-risk population as they progress from just genetic risk through the development of autoantibodies to actual T1D diagnosis. We've learned so much about the progression to T1D diagnosis through these studies, and we've been able to reduce the risk of DKA at diagnosis through these studies. These studies have been able to recruit patients into clinical trials and have actually led to a lot of the findings that we're we're dependent on today that we'll talk about a little later for our disease-modifying therapies. Importantly, they continue to support T1D families as they go through what might be a difficult risk diagnosis for them, giving them both educational support, personal support, and medical support, because screening alone is not sufficient to reduce your risk of DKA or improve your health. You need to be monitored. You need to be working with a doctor through your risk diagnosis to find out when you're transitioning from stage 1 to stage 2 to stage 3. We're also identifying what are the barriers that are preventing people from participating in clinical research-based screening studies like these. What are preventing our non-European, non-Caucasian populations from participating? And we're trying to improve the enrollment from Hispanic populations, African-American populations, non-white demographics, to better improve the diversity of people being screened. And as we'll talk about later, these programs have been crucial to the development of our disease-modifying therapies. In more recent years, we've been looking at the importance of general population screening. Again, I remind you, 93% of people who are diagnosed with T1D do not have a family history of disease. So while important, these familial-based programs that I just talked about do not capture the majority of people who are at risk for the disease. So we need to be able to make T1D risk screening available to people in the general population. Programs like ASK, FRIDA, TEDDY, and DIP have done just this. They've shown that these programs are feasible, can be cost-effective, and can impart a real public health benefit by reducing rates of DKA diagnosis, by informing and educating patients about their risk of T1D, and by recruiting people into clinical trials. We know that the health outcomes from people who participate in these programs are dramatically improved compared to those who don't have access to screening and monitoring. So now what JDRF is doing is we're looking at how to expand these programs and capitalizing on what we've learned from ASK, FRIDA, TEDDY, and DIP, but also what we've learned from TrialNet and the other familial-based screening programs. So with what we've learned from these research-based screening programs, JDRF launched a little over a year ago a community-based real-world screening program called T1 Detect. T1 Detect is actually an education, screening, and awareness program. Our goal here is to educate people about T1D and T1D risk so that we can teach people really in a general population why it's important to get screened for T1D. That's the first step. People need to know why it's important before they're going to go and get screened. So we're teaching both people in the community, but also their healthcare providers, because many healthcare providers don't know why it's important for a person to get screened for T1D. So we need to teach those people first before we even provide them access to a screening tool, which we're already doing. So if you're interested in our T1 Detect program, you or your loved ones or your friends can go to JDRF's website, find T1 Detect, and you can actually order a screening test. The test costs about $55. If you can't afford that, we can provide you with an assistance fund that will bring the cost down to $10. The test gets sent to you at home. You do a simple blood test, you spot your blood on a card, send it to the testing company, and they'll send you back your results for the major autoantibodies that detect whether or not you have autoimmunity for T1D. Those results will need to be confirmed, but if confirmed, that will tell you if you're at risk for T1D and whether or not you need to be monitored more closely for your risk. So we're really proud of this program, and we really hope that you and your friends can teach your other friends about T1D risk and T1D screening. So let's move on to actually treating and curing the disease with disease-modifying therapies. How are we going to turn off your autoimmune cells? How are we going to improve the survival of or regrow your beta cells. There really are a handful of key players in this field, and they come down to a couple different cell types in your body. The first and foremost is really your insulin-producing beta cell. They produce insulin, but during the course of T1D, for various reasons, they become stressed, they become not healthy, and they become more attractive to the immune cells, more easily killed by immune cells floating around your body. So we're looking for therapies that are going to improve their survival, number, and function. In fact, we've found therapies that can improve the survival of the human beta cell and have them in clinical testing right now. Verapamil, the blood pressure drug, is one such example, and that's now in multiple clinical trials testing its efficacy to improve beta cell function and survival after diagnosis with T1D. Then there's another type of cell in your body, in your immune cells, called T effector cells. Their job is to fight threats from bacteria and viruses and get rid of them for your body to keep you healthy. However, in T1D, in autoimmunity, they turn their attention to the cells in your pancreas, to your beta cells. There's too many of these T effector cells floating around, and their attention is turned to the wrong cell. They start killing your beta cells. So what we're trying to do is we're trying to find therapies that can disable them, either disable those T effector cells or get rid of them. And in fact, teplizumab is a great example of this. Teplizumab has been tested in multiple stages of type 1 diabetes, and right now is sitting and waiting with the FDA for, we hope, an approval soon as a drug to delay the onset of clinical diagnosis of type 1 diabetes in the at-risk community. Another important player in type 1 diabetes are the T regulatory cells. Their job is a peacekeeper. They reduce the aggressive abilities of the T effector cells, another immune cell population. They try and keep them calm. Now, in type 1 diabetes, there's not enough of these peacekeeper T regulatory cells around. There's not enough of them, and they're not very good at their job. So we're looking for therapies here that's really going to enhance their function, that's going to make them either have more of them floating around or more powerful. And one way to do this is through antigen-specific immunotherapies. And right now we have some of those being tested. We're trying to bolster the number of T regulatory cells floating around. Many clinical trials have been done here with the pre-point studies, with oral insulin studies. And in fact, Novo Nordisk is running a clinical trial now called Topple T1D, where they are using a DNA vaccine to produce antigen-specific T regulatory cells for insulin. These will be peacekeeper cells that when they see cells that want to attack insulin-producing beta cells, they'll tell them to calm down. Lastly, there are these immune deviator cells. These are just immune cells that support the function of the other T cells, the T effector cells and the T regulatory cells. Now, these cells are either in T1D not really good at their job, or they've chosen the wrong side. They're supporting the killer T cells instead of the peacemaker cells. So we want to find ways to bias them towards supporting the peacemaker cells. And we can do this with therapies like low-dose IL-2 or other cytokine therapies where we can bias their activity towards supporting the good guys. And we'll talk about some more of those disease-modifying therapies a little later. So let's get into some of the clinical trials that are going on right now. One that I'm personally interested in, because I've been working with this one myself for the last couple of years, is the BANDIT trial that launched last year in Australia. The BANDIT trial is using a class of drug called a JAK inhibitor. A JAK inhibitor is a type of immunosuppressive drug that's being used in multiple autoimmune diseases right now. It's approved for multiple autoimmune diseases right now. Now, one autoimmune disease is not terribly different from any other autoimmune disease. Type 1 diabetes is an autoimmune disease. So what we're doing is we're capitalizing on what's being used in diseases like rheumatoid arthritis or multiple sclerosis. And we funded research with an academic investigator in Australia named Helen Thomas to prove that this class of drug, JAK inhibitors, could be effective in type 1 diabetes. And Dr. Thomas found that that was in fact true. And when she found out that was true, people at JDRF, including myself, worked really hard to convince pharmaceutical companies to agree to give us their drugs, that we could use those drugs in clinical trials. And with support from Drs. Thomas Kay and Helen Thomas in Australia and people at JDRF, we were able to convince colleagues at Eli Lilly to give us their drug baricitinib for a clinical trial that launched in December last year. So this is the first ever clinical trial using a JAK inhibitor in the recent onset population to see if it can keep beta cells alive and keep the immune system at bay. And we're hoping that this will read out, right now it's planned that this will read out in 2023. So keep your eyes peeled. Also, since this trial is going in Australia, we want to see trials like this happen in the United States. We should see that happen next year. So ATG is a drug called anti-thymocyte globulin. And it's a type of antibody that specifically removes T cells from your circulation. Now remember, I talked about T cells before. There's T effector cells, which are the ones that are killing your beta cells. And then there's also T regulatory cells, the good guys. Well, it's been found that anti-thymocyte globulin can specifically remove those bad guy T effector cells from your circulation. Now, JDRF has been funding the preclinical research around this for a number of years, and in fact, also helped fund the clinical trials around this with Mike Haller down in Florida. It was found that if you treat people after diagnosis with ATG, it not only preserves beta cell function, preserves the ability of your pancreatic insulin-producing beta cells to keep producing insulin. It reduces your HbA1c, it keeps you producing insulin, and it reduces your need for exogenous insulin, for dosing insulin. And we can see this on the right here, that when we look at the ability of people to produce insulin as read by C-peptide, people in the red line, those are people who weren't on the drug, keep declining over the course of 2 years after treatment. But people in the blue line, people who are on ATG, they had preserved their ability to keep producing insulin. So because of these important results, ATG is now being studied in the context of the Anodia Consortium in Europe, and also is ready to be studied here in the United States in other clinical trials consortium as a preventative, as a way to delay the onset of type 1 diabetes. It's a drug from Sanofi, so we're hoping to see this move through development as well. And I mentioned this guy before. I mentioned verapamil before. Verapamil is a blood pressure drug that the work of Dr. Anath Shalev at the University of Alabama at Birmingham has shown that this blood pressure drug has the ability to reduce beta cell stress. Beta cells get stressed during type 1 diabetes. And when beta cells get stressed, they can't function as well as they're supposed to. They can't produce the insulin they need. And in fact, if they continue to be stressed, they end up dying. So Dr. Shalev showed that this drug can preserve beta cell function in animal models of type 1 diabetes. And because of those important findings, we worked with her to do a clinical study that's shown here on the right, that if you give this drug to people after they're diagnosed with type 1 diabetes, you can not only preserve their beta cell function, but you can almost keep them flatlined. They don't lose any ability to produce insulin over the course of a year. Again, we're looking at the red line here are the people who were treated with verapamil. And you can see from 0 to 3 months, they actually have a little bit of a boost in their ability to produce insulin that then declines a little over the course of the next 9 months. But people who are on placebo just continue to decline. So because of these important findings, verapamil is being utilized now in multiple clinical studies. in the United States here, there's the CLEVER study, which is combining a pump and device system, an artificial pancreas system, with verapamil in very young children to see if that can preserve beta cell function better after diagnosis. And then in Europe, the VERA T1D study, to repeat the findings of Dr. Anashalev in many more people to see if the results hold true to a wider population. So I'm glad to have had the opportunity to show you some more details about a couple of the clinical trials we're working on, but what I want to point out here is they're only a small subset of all the trials that are going on in type 1 diabetes in all the stages of the disease. From the earliest stages at stage 1, when a person just has signs of autoimmunity and autoantibodies but no symptoms, there are drugs like hydroxychloroquine, abatacept, and oral insulin that have or are being tested in that population to prevent their further development of autoimmunity, to prevent their further need for exogenous insulin, and hopefully prevent them from developing any need for insulin. In stage 2, teplizumab, a drug I hope you've heard a lot about and will continue to hear about, that's looking to stop people from progressing from stage 2 when they do have metabolic dysfunction, but they don't have symptoms, to prevent them from going forward into the disease to when they would need insulin. And we know from clinical trials that this is working. There are people who are living right now on teplizumab or after being dosed with teplizumab who are still type 1 diabetes free after 3 years of being on the drug or 3 years after treatment that should have been diagnosed already based on what we know from T1D risk progression, and they haven't been diagnosed yet. So again, we're hoping to see an FDA approval on this in the coming year. Marjana will talk about this a little more later. And then we go into stage 3 disease, where teplizumab, verapamil, iscalimab, impact, there's just a host of disease-modifying therapies being tested here, both beta cell drugs, immune drugs that might target both. Things like L-lactis, which is a bacteria that's been genetically modified to secrete a good protein that will help foster the development of your good T cells, your T regulatory cells, as well as an antigen. So it's sort of a bacteria that's delivering an antigen-specific immunotherapy to your gut. And JDRF has been a leader in all of these. I want to leave you with that before I transition over to Marjana, but JDRF has been a leader in all of these. We work with the investigators to help them put together preclinical data packages that will hold their water when we start talking to the different companies that own these drugs. And we work with the investigator and the companies to convince those companies' senior management that T1D is the right path forward, that we can help them access clinical consortia like TrialNet, like Inodia, like the Australian CRN, to test these in T1D populations efficiently, that we have staff members like Marjana and the rest of her team that know what the regulatory path is like for T1D. We have the resources that can get these from the very, very beginning of just the ideation of a DMT, a disease-modifying therapy, all the way to the patient's hands. So it's often a long process, but what we're seeing now is, I think, a really fruitful and a lot of successes coming to fore right now. So I think I'll hand off now to my friend Marjana and let her talk about how we're handling the regulatory considerations for how do we get the clinical data, the really positive clinical data for a lot of these to our regulators for an approval, and how do we work with other policy considerations around drugs like these. So thanks everyone, and I'll hand it over to Mariana.
Thanks so much, Frank. Hi, everybody. I'm Marjana, and it's great to be with you all today. So I'm going to go over some policy considerations. I'll start with regulatory considerations and then move a little bit into the health policy and our efforts around screening. So as you can see here from this slide, and I imagine many of you have seen this before, and as Frank talked about, one of the strengths of JDRF is working across the pipeline. And this is the pipeline that we often refer to, and advocacy is really critical and important to JDRF's mission. And our ultimate goal here is better outcomes for people with T1D. And we work towards that by working, as I just mentioned, across the development pipeline so that we can get these therapies into the hands of people with T1D and those at risk for T1D as quickly as possible. And to help achieve this goal, you heard about Frank and all the research that we fund and a really strong research team who funds research in the discovery and the translational space. Down in the advocacy office in DC, our government relations team really focuses on ensuring that there's strong federal commitment to T1D research funding through the Special Diabetes Program. And then for the regulatory team, which I'm a member of, we want to make sure that there's reasonable regulatory pathways for the research and for the cures and prevention and improving lives therapies that we're working on. And then there's the health policy team that works to increase healthcare access and adoption of those T1Ds. Ultimately, again, the goal being better outcomes for everyone with T1D. So we've been working on regulatory pathways for T1D therapies and really clarifying and shaping the policies at the FDA and regulators around the world. An important part of that is outcomes or endpoints that can be used in clinical trials for new therapies. Hemoglobin A1C, which I know you all are familiar with from clinical care, is also the most commonly used and the primary efficacy outcome measure that's used in the majority of trials looking at T1D. But as the types of therapies, just as Frank was talking about, are evolving, where we're intervening in earlier stages of the disease, and we're looking at markers of autoimmunity and how we're modifying the immune aspects of this disease. And as the science evolves, we want to make sure that developers have additional outcomes available for them to use. So we're working on several different fronts, and I'll get into the details of these in the coming slides. We're working with FDA, obviously, but we also need to work with industry and researchers to establish that these additional outcomes are getting incorporated into their development program and in the decision-making. Outcomes like those that are measured by continuous glucose monitors. Namely there, we're talking about what time in range, but there are other metrics that could be really important here in earlier stages of the disease as well. The islet autoantibodies and identifying those people at risk as part of the screening programs that we just talked about and incorporating those into the regulatory decision-making process. And the use of C-peptide, which is a marker for endogenous insulin production. How much of your own insulin are you making? And that's a really important measure in trials for new onset type 1 diabetes. Having these outcomes available for use in development holds the promise of faster and more targeted clinical trials. So let me get into a little bit more detail about the use of CGM metrics in drug development. We've seen in the last several years, as I know all of you are aware, really the explosion of the use of continuous glucose monitors and improvements in what they're able to do, right? We are now in a place where we have artificial pancreas systems on the market and the ability to use CGM data to make insulin dosing decisions, which not many years ago, CGMs were at that point what was called adjunctive, meaning you needed to confirm what was happening or what that CGM was telling you with a test from your blood glucose meter before you made a treatment decision. And the technology has advanced and evolved in a way that now they're part of artificial pancreas systems and running algorithms and being used to make treatment decisions. And even without an AP system, if you're using multiple daily injections, you can use your CGM now and some of the more newer CGMs, you can use some of those in order to make treatment decisions. What we've been doing is advocating to both academia and industry and the regulators that those CGM metrics and measurements of those CGM metrics are included in research and development programs. Because at the end of the day, right, this is what patients and caregivers are using day in and day out, is that number to make a treatment decision. And we want to, better understand hemoglobin A1C, has its limitations. It's an important marker. It is still an important marker of your risk for long-term complications. Having the CGM data, it gives a quality to what's going on with A1C, and it helps patients and caregivers understand day in and day out, what's happening with the treatments that they're using. One of the things that we do know is that larger data sets are going to be needed in order to answer questions around the clinical meaningfulness of CGM metrics, in order to be able to use them as an efficacy endpoint in studies and for approval of drugs. What I mean when I say that in clinical meaningfulness, so what we know is hemoglobin A1C is a surrogate for your risk of long-term complications. And we got that information from the DCCT studies, if you'll recall. What we need to establish for CGM metrics as well is, why are they clinically meaningful? And what that means in terms of regulator speak is, how does it affect how you either feel, function, or survive? And so we are working actively to encourage academic trials and industry trials in order to gather those large data sets of information that we're going to need in order to establish the clinical meaningfulness of CGM. What we've also seen, which may seem subtle, but I recall the days of seeing FDA's position on CGM metrics being exploratory, right? That they wouldn't necessarily encourage sponsors to collect it. They were more than, the sponsors could, they didn't have to get FDA, if you will, to collect those, but FDA's thinking at that time too was that, well, these are just exploratory endpoints. We really don't know much about this technology or what actually these CGM metrics data means. What I'm seeing now, which is a step in the right direction and tells us that the FDA is thinking about the drug center in particular, how are we going to use this data in assessing safety and efficacy for drug therapies and interventions? They're not the group who reviews CGMs and approves them. That's the CDRH group, the Devices Center at the FDA. So when we see the drug center now actively encouraging and asking sponsors to collect those CGM metrics in their drug development programs, it tells us and it signals to us that they're looking for those large data sets in order to validate the clinical meaningfulness as well. And we also had another movement, which is really positive. And as many of you may recall, several years ago, we launched the Outcomes Beyond Hemoglobin A1C program, right? And this was coming on the heels of understanding, right, that there are metrics that are important for people with type 1 diabetes and their caregivers. And what we saw too was that many of those metrics had various definitions. If you looked at different development programs or different research studies, there was an inconsistency in how those metrics were defined. So what we did was we brought the community together and built consensus on one, identifying those clinically meaningful outcome measures beyond hemoglobin A1C, and then putting standardized definitions to them. That then was endorsed and published and adopted into ADA's clinical guidelines. One of the important works was around hypoglycemia and what's called level 2 hypoglycemia. This is blood sugars that are less than 54. Up until very recently, what the FDA recognized as clinically meaningful was severe hypoglycemic events. But we know that level 2 events are really important to patients and caregivers, that it does impact their ability to function and how they feel. And so we see recognition now from the FDA that level 2 is clinically meaningful. And what we're working on to ensure is that using, where we're at today is that those can be measured using a blood glucose meter in research and development programs. And what we want to step towards is, that CGMs could be used to measure level 2 hypoglycemia. But again, we're seeing an evolution of this now allows for companies and sponsors and researchers to look at events of level 2 hypoglycemia, and improvements in those could lead to approval of a therapy, because reduction in those events would be considered clinically meaningful. I also want to mention here too that we, as part of the Outcomes Beyond A1C program, had launched a patient preference study. And that was published about 2 years ago, where we identified what patients and caregivers preferred in terms of outcomes beyond A1C. And not surprisingly, what we saw was that patients and their caregivers prioritized in the frequency and severity of hypoglycemic events and hyperglycemic events. And in that study, we intentionally did not include severe hypoglycemic events because we knew that those were already clinically meaningful. We included the level 2 events, which was less than 54, and the level 1 events that were less than 70. And patients and caregivers did identify that those were important to them. And that's what they preferred in terms of outcomes, looking at those being hemoglobin A1C. Now, let me move into biomarker qualification of autoantibodies and why this is really important. As Frank mentioned, screening efforts for JDRF are really important in identifying people who are at risk for type 1 diabetes. JDRF brought together the community back in 2014 to talk about staging the early stages of type 1 diabetes. So before you became insulin independent, because as Frank just mentioned, we know that this process can sometimes start many, many years before you actually become insulin dependent. And how do we identify those people? And what we knew from the TrialNet studies and the screening studies that had been going on at that time is that if you have 2 or more islet autoantibodies and that you're starting to show signs of dysglycemia, you're at a significant risk for developing type 1 diabetes. And what we would say and show is that it wasn't a matter of if, but a matter of when. Now, what we did when we brought the community together to establish this consensus and the staging, the regulators were there. We invited the FDA. And one of the things that we heard from them was that they had seen the publications, but they had not seen the patient level data that would allow them to make a decision or to include islet autoantibodies as enrichment markers for prevention studies in type 1 diabetes. So what we did is we found CPATH, who's a nonprofit organization who focuses on bringing data sets together for regulatory qualification of biomarkers. And that's what these islet autoantibodies are considered. And so that consortium was launched 3 years ago. We have currently the data is being reviewed by both the FDA and the EMA that would qualify the islet autoantibodies as risk markers, enrichment markers for prevention studies in type 1 diabetes. And so, this is exciting progress. We're expecting both FDA and EMA in the coming year here to have decisions that recognizing the islet autoantibodies as enrichment biomarkers in prevention studies for type 1 diabetes. Moving on to new onset type 1 diabetes. As many of you are aware, and as Frank discussed, there's been much research in folks with new onset type 1 diabetes and disease modifying therapies. And CPATH recently brought together the community. JDRF was a member of the planning committee that included both the FDA and EMA and research experts to talk about the design of those clinical trials in new onset type 1 diabetes. We worked closely with those research experts and in industry to ensure that we were providing a unified and consistent marker that C-peptide is the right measurement in these studies, that it is a measurement of endogenous beta cell function, and the data and the evidence to support that. One of the things that we also identified as part of our efforts leading up to this meeting, and we'll talk about JDRF's involvement here in the coming slides, is that the consensus around C-peptide is actually pretty old. It's back from like 2002. And so, there's been a lot of research that's come out in this space since then. And so we've been working with a small group of key opinion leaders to update that consensus on C-peptide and provide updated information. So where that consensus paper is in its final stages of review, and we hope to see that published in the coming year as well. One thing I'll mention here in the new onset space as well is that one of the things that FDA said at that meeting, which is a step in the right direction that we had not heard before, is that they recognized that C-peptide was what they call a reasonably likely surrogate endpoint. And what that means is that allows those therapies that are studying that space using C-peptide to use what's called the accelerated approval pathway. And what that means is that you don't have to confirm the clinical benefit pre-marketing. I mean, you can get your drug approved without having that confirmation of clinical benefit in the pre-market setting and actually work on that clinical benefit confirmation in the post-marketing setting. So what that does is accelerated approval, commonly used in oncology space and cancer studies, could allow for smaller studies and quicker approval of therapies in the new onset space. Now, update on teplizumab that Frank talked about a little bit earlier, and a really key therapy in terms of disease-modifying therapies. For those of you who aren't aware, FDA had granted breakthrough therapy designation to teplizumab for the work that was being presented in stage 2 of the disease and those who are at high risk for developing type 1 diabetes. And what that means is that clinical data to date supports that this is addressing a significant unmet need, and that therapies that get breakthrough therapy designation get what I'll call special attention from the FDA. Their senior leadership is highly involved in working with the sponsor to accelerate the review and development pathway for therapies that have breakthrough therapy designation. So this was exciting news to see for teplizumab. On May 27th, there was an advisory committee meeting that happened about this application, where the committee voted to recommend that FDA approve this therapy. During that committee meeting, there is the open public hearing session where patients and the community can come and talk about their perspectives on what's being discussed at the FDA. Aaron Kowalski from JDRF, our CEO, gave compelling testimony at that advisory committee meeting, really underscoring that a 2-year delay in the onset of type 1 diabetes is clinically meaningful. And resoundingly, you heard the patient community really step up and also reinforce that. Well, what I'll also say is that in reviewing the briefing materials, this is the materials that the company and the FDA puts together in preparation for this meeting that the advisory committee gets to review. I saw for the first time a description of the unmet need by FDA around T1D that I'd not seen before. And I give credit to JDRF and our work for reminding the agency and continuing to engage with them about the need for cures therapies in type 1 diabetes. What I also saw in there, which I hadn't seen before either, was a recognition of the staging criteria. The one where they had said, we haven't seen this data and we really can't make decisions on this. We saw that information in the briefing document, which was really important. Now, Prevention Bio did receive a complete response letter from the FDA on July 2nd, and what that meant was that at this time, the FDA wasn't able to make a decision on approval. Now, what Prevention, the company who is the sponsor of teplizumab, told us in their public press release about this complete response letter was that there wasn't identified any issues with the clinical data. And I say this because that's really significant. If you take a look back at the stage 2 trial, this TN10 trial that this BLA is based off of, it was a very small study. And it was a small study in terms of the number of people who actually received teplizumab. But there was a recognition that these patients are really hard to find, and that there's a significant unmet need there. And FDA was willing to consider additional data from the new onset space for teplizumab in order to support approval. So really sort of profound advances, if you will, in recognition of the unmet need and what could be considered in order to support approval for teplizumab. While we're disappointed with the CRL, I think what it looks like the FDA is looking for more information on is around the comparability. Teplizumab is an older drug and is common for new manufacturers to come into place. And so the sponsor is working with the FDA closely in order to get that additional information to them. And we're hoping to see that approval come through in the next year as well. And if you're looking for more information, you can find that on our blog, the JDRF blog from July 13th. And last, again, this is just a timeline going over much of what I've just shared with you all, starting back in 2014, where we hosted a consensus workshop to help recognize and define those early stages of type 1 diabetes, to the publication and endorsement in 2015 of those early stages from in JAMA and by the leading clinician organizations. The 2017 consortium being launched for the islet autoantibody qualification, and FDA accepting and supporting that biomarker qualification in 2018 from the FDA, and here most recently in 2020 from the EMA. And then, teplizumab getting that breakthrough therapy designation from the FDA, and now here in 2021, while we do have that complete response letter, I think we're optimistic that the sponsoring FDA can and will work through those issues. And seeing the progression of FDA's thinking for disease-modifying therapies based on this last workshop here in June, all leading toward what we hope and what we know will be disease-modifying therapies for type 1 diabetes. Let me move now into our health policy area. As I mentioned before, making sure that we have coverage of interventions and screening programs for type 1 diabetes, as well as clinical adoption, is really necessary and a key component for leading to better outcomes for people with type 1 diabetes. Now, we want to make sure that our screening programs and monitoring are part of clinical preventative services. So where we're at today is that screening is not part of routine preventative services, and it's paid for only sporadically in the US and in other countries. And there's limited provider and public knowledge and awareness. Where we want to go and what we're working to do is to make sure that screening for type 1 diabetes is routine for all patients, and that is paid for by the public and private players, and really increasing the knowledge and providing education of the risk of T1D among providers and patients. And as Frank mentioned, the majority of people who are at risk and get diagnosed don't have a family connection. Now, our approach to doing this and how we're doing this is, one, educate. Awareness is key and important here. Educating the key provider stakeholders. Also educating the top 25 health plans. If we could go to the next slide, please. And also educating the US Centers for Medicare and Medicaid Services. In terms of coverage, we want to ensure that screening and monitoring, if needed, is reimbursed for relatives of people with T1D, but that screening and monitoring is also covered and reimbursed for the general population. Recommendations that we're pursuing include inclusion in ADA and other clinical guidelines for general population screening for T1D, and inclusion of screening in the American Academy of Pediatrics Bright Futures program, as well as a positive rating by the United States Preventative Services Task Force. In addition to this, we're assessing, and I am working closely with the health policy team and the research team to make sure that we can secure access to these disease-modifying therapies once they're available. We want to make sure that there's broad commercial insurance coverage, and government programs that cover those DMTs, that they're accessible at an affordable out-of-pocket cost, and limiting the use of prior authorization in this stage. And how we're working on that is engaging insurers, employers, pharmacy benefit managers to raise awareness. And really the attention here will also increase more, and there'll be more opportunities as we get the first disease-modifying therapy approved by the FDA. With that, I'm going to turn it back over to Frank.
Thanks, Marjana. So I hope you have a better understanding now of how our research-based activities, our discovering new disease-modifying therapies-based activities are leading into the work of Marjana and our team, and how she and her team are able to work with the regulators and the scientists to bring these drugs into the hands of people with type 1 diabetes. Just to speak for a minute right now on trials that are available to you and your loved ones. First, please go to JDRF.org and find our clinical trial connector tool. It will help you find in real time, given your geography, your age, your HbA1c, your autoantibody status, what trials you can participate in right now, because participating in clinical research is how we can move these ideas, these potential disease-modifying therapies into the hands of people faster. So it is as much your responsibility as it is my responsibility to help support this research. Some of the studies that are going on, we mentioned teplizumab. This is a prevention study, but also a study in the newly diagnosed, in the PROTECT study. The BANDIT study in Australia is on the JAK inhibitors. The TOPPLE study is a study run by Novo Nordisk that's looking at a DNA vaccine for insulin to recreate your immune tolerance to beta cell proteins, in this case, insulin, to make your peacemaker cells more potent at stopping any immune attack on those insulin-producing beta cells. Ustekinumab is a drug that will stop the activity of two important inflammatory cytokines, which are, through JDRF-funded research and other research, has been shown to impact the progression of type 1 diabetes and the activity of the disease. So we want to see if ustekinumab can slow the progression of the disease after diagnosis. Moving on to our Enodia platform in Europe, there's 4 trials going on there right now, with more set to start next year. For Rapamil, I mentioned this is our beta cell survival agent. MELD ATG, I mentioned ATG before. This is to deplete the killer T cells. Iscalimab is a Novartis drug that is repurposed from the transplant setting, that is hoping to slow the loss of beta cells in newly diagnosed adults and adolescents by stopping the immune attack on those beta cells. And then Impact is using a type of drug called an imitope to, again, stop the bad T cells from killing your beta cells. And then again, here's our CLEVER study, which is super innovative. Jen McVean is one of the lead PIs, is bringing devices and drugs together in the same trial. So we're hoping to start seeing results from these in the next couple of years and seeing the launch of more in the next couple of years. So look around and see if any of these trials resonate with you, and maybe you might want to participate in them. So today, I hope you appreciate from what Marjana spoke about and what I spoke about, what are the critical needs to get to cures involving disease-modifying therapies for type 1 diabetes, how our screening program is expanding and needs to expand to help, identify individuals who might benefit from disease-modifying therapies when they're approved, to help improve the health of people as they get diagnosed with T1D, reducing the incidences of DKA, how they need to expand to identify people who aren't normally being identified through some of the other research programs that are going on. We need to be able to identify these people in the general population so that we can reduce the rates of DKA in the world, so that we can improve people's health in the world. And then, our real world community screening program, T1 Detect, is just one way that we're trying to offer this risk screening program to people in the general population. We are really excited about what's happening in the disease-modifying therapies world right now. I hope you felt my enthusiasm. There are a lot of trials running. There are a lot of trials that have shown really exciting data. We've mentioned teplizumab a lot, but that's just one of them. There's ones that will read out this year and next year and the following year. And these are based on 10, 20 years of JDRF funded research, on the efforts of JDRF staff, on the efforts of T1D expert scientists that we've been funding, based on your efforts to help us fundraise. So it's important that we maintain that that enthusiasm, we maintain that accelerated path forward, because we need these multiple shots on goals. Not one drug is not going to be enough. Remember, we're going to need some beta cell drugs. We're going to need some immune drugs to really tackle this disease. So, JDRF is pivotal to the successes here. Our volunteer staff, our scientific staff, our regulatory staff, our advocacy staff, our scientific partners, our nonprofit partners. What we at JDRF can do is we can bring them underneath the one tent towards the one mission that we agree on, getting cures for T1D. And so it's our role to make sure that everyone, all the right people are at the table, and all those people are moving in the right direction. Please pay attention to the clinical trials recruiting in your area. Hopefully that'll continue to accelerate as we come out of COVID. It's amazing that even in COVID, clinical trials kept recruiting. The Bandit trial launched in the middle of COVID and kept recruiting. So as we come out of this COVID pandemic, you'll see an increased acceleration of more clinical trials. And we can't thank enough JDRF advocacy in their efforts working with the FDA to make sure the FDA is aware of what the needs are in the T1D community. They know what the science is right now, and we provide them that data to say, this is how we think about the disease. This is the data that's available on staging. This is the data that's available on C-peptide as an endpoint. This is the data that's coming on the use of CGMs in clinical trials. What do you, FDA, recommend we do in our next clinical trial so that when we bring that data to you in the future, you'll look at it favorably? Without JDRF working there, we wouldn't have that dialogue to make our future clinical trials more efficient and more likely for success. So next month, we'll be looking more into the preclinical space. So moving back into the lab, and what are those technologies and potential therapies that are being developed in the lab that might be the next potential cures for T1D that end up in clinical testing. So with that, I can't thank you enough for tuning in. I can't thank Marjana enough for speaking today. And I look forward to talking to you live and answering your questions next week.